It, hello, it's my great pleasure today to welcome all of you to the Harvard Medical School PhD hooding ceremony for 2023. Welcome graduates, welcome families, faculty, program leaders, program administrators. Welcome to Dean Dench and the team from GSAS. I am Roz Siegel, the Dean for Graduate Education at HMS and co-director of Harvard Integrated Life Sciences, together with my friend and colleague, Victoria D'Souza. It is really such a great pleasure to be able to come together and celebrate our wonderful and talented colleagues, the new graduates. In addition to the usual and very difficult path of carrying out PhD research, they've had to put up with all the impediments due to COVID-19. But they have persevered and have carried out important and truly groundbreaking research. We now have the great opportunity to cheer them as they move on to new endeavors in life sciences. So this will be the first chance to cheer them. So cheer. I would like now to introduce our student speaker, the newly minted Dr. Isla Bastille. Isla did her undergraduate studies at Cornell, where she double majored in biology and psychology, graduating magna cum laude. While an undergraduate, she worked both on the evolution of visual circuitry in birds and on methods of visualizing active neurons of the spinal cord in moving mice. In graduate school, she carried out studies on the auditory system in the laboratory of Dr. Lisa Goodrich. Her absolutely beautiful and rigorous analyses identified the ways in which transcription factors together code for differentiation, morphology, and circuitry of spiral ganglion neurons that enable us all to hear. In addition to her beautiful work in the lab, during the pandemic, Isla developed a wonderful series of educational scientific videos. She was the president of the Underrepresented Scholars in Neuroscience, and she initiated a new student mentoring program. Her work and achievements have been recognized by a Gilliam Fellowship from HHMI, and she's received several times the Photo of the Year from the Harvard Brain Initiative. After graduation, she plans to use her skills in bioinformatics to advance sustainability research. Isla, it's great to welcome you here.
Thank you for the introduction, Roz, and welcome, graduates, guests, faculty, and staff. It is truly my honor to have been chosen to speak on this momentous occasion. I'd like to start off by describing an experience that I know will resonate with many of my fellow graduates. It's December of my first year in graduate school, and I'm wading through 2 feet of snow to get to my 8:30 a.m. class. Meanwhile, classmates from California are dropping like flies as they reckon with the concept of winter and the lack of decent burritos in Boston. After a 3-hour lecture, of which I will remember about 2%, I have lunch with my intimidatingly intelligent classmates, where we launch into a heated debate on the utility of this hot new method, single-cell RNA sequencing. I run to my rotation lab, where I drop my gel on the floor, ruining weeks of work, and walk back home, defeated in now 3 feet of snow, to my crooked Brookline apartment, where I settle in for a Trader Joe's frozen meal and Google, what even is single-cell RNA sequencing? About 3 sentences into the seminal paper, I fall asleep In the morning, I wake up and give it all another go, stopping at that glistening Harvard Medical School sign to ask, what am I doing here? 7 years have passed, and I'm glad to say I have some answers. Today, I hope to reflect on how we have braved a tumultuous, often uncomfortable learning process to become indispensably talented at understanding ourselves, others, and the world around us. I propose that these talents are not only sufficient, but critically necessary as we become scientific leaders and shape our experience on this planet. Shortly after the scene I previously described, I passed through another universal graduate school experience, crippling procrastination about a grant. But as I was sitting in yet another early class, Dr. Siegel showed us a video depicting the intricate inner machinations of a brief moment in the life of a single cell. It dawned on me that these complex events were being seamlessly executed all the time in the roughly 40 trillion cells in my body. Suddenly, I didn't feel so lazy anymore. Instead, I felt pure awe that I was embarking on a journey to contribute to understanding the myriad of tiny miracles that have made it possible for me to be alive. As life scientists, we have studied the processes that make life possible. From the improbable collision of ancient molecules to this very moment, this gathering with me standing before you, trying to communicate how this immeasurable accomplishment makes us all feel. We have all contributed to the deep understanding of ourselves. And while I can't say that this awe has always rescued me from procrastination, I hope that this anecdote reminds us that from our brilliant discoveries down to our hardworking kinesin molecules, we're all literally awe-inspiring. And that in times of doubt and procrastination, it helps to stay curious about the many tiny miracles within ourselves. And what is fascinating to me is not only this little orb of curiosity we've carried with us in this process, but the ubiquitous phenomenon of honing our capacity to share this curiosity with others. We've all passed through the awkward, almost date-like experience of rotating and choosing a lab, and ultimately coming around to prepare our first lab meetings. And let me tell you right now, the great majority of our first lab meetings were really bad. I think the diplomas are nice and all, but if you want to feel really accomplished on any given day, please open the first scientific presentation you ever made in grad school. No, really, trust me, you'll realize how far we've come at being able to communicate extremely complicated concepts to each other. Compare it to your defense, for example, where you brought together so many of the people you respect scientifically, as well as your loved ones, just to say, hey, come look at this amazing, complex thing I've discovered. Through conferences, lectures, papers, reviews, posters, beer hours, hallway chats, and lunches, it has become secondary for us to share our work and feedback. I've realized that this is actually a veiled way of saying, let's be in awe of life together. In this way, we've become bigger than the sum of our parts. We've integrated into robust networks of knowledge simply by sharing our curiosities, like children in a backyard gathering around to marvel at a firefly cupped between tiny hands. And it's not just the graduates that can experience this joy of discovery and wonder. Parents, families, and loved ones, I implore you to be reciprocally curious about the portal of knowledge that your beloved graduate has become. It is a precious experience to be able to share what we know with you, too. You might not understand everything we say, but here's the dirty secret. Not too long ago, we didn't either. Worst case, you'll understand the world and your graduate just a little better. I recently told my grandmother that we are made up of trillions of teeny bubble-like machines that work tirelessly to keep us alive. Later that evening, I caught her looking at her hands as if she was seeing them for the first time. Unsurprisingly, this capacity to grow, learn, and communicate has bled into our social lives, where we have stitched ourselves together through student groups and networks to stand up and fight against gaps in knowledge and justice in our day-to-day lives. Whether it be racism, sexism, cancer, or a global pandemic, I am so inspired to be embedded in this web of people who will persistently reach out to each other to make this world better. And we've done this despite navigating harrowing personal obstacles, setbacks, and grief that at times seemed insufferably magnified or muted by immense collective grief. We've personally experienced or supported classmates through mental and physical health struggles. We've lost classmates or loved ones, and at times almost even lost pieces of ourselves. We have become very, very close friends with failure. We've had to admit defeat. We've had to ask ourselves, what am I doing here? And yes, grad school is rife with inordinately difficult obstacles and failures, but this also makes it full of firsts. The first time you collected a single data point, the first time you thought of an experiment on your own, the first time you realize that no one says, "Eureka," and instead says, for years at a time. The first time you asked for help, the first time you overcame a failure, the first time you helped someone else overcome a failure, the first time you read a paper and actually understood most of it, the first time you realized you discovered something, the first time you realized you could actually do this, the first time you shared your work, the first time you realized you were a real scientist and not a stack of koalas in a lab coat. The first time you could tell a younger student it was going to be okay and believe yourself when you said it. Grad school is bespeckled with moments where you can tangibly pinpoint exactly how you were growing and overcoming and what that felt like. In fact, I argue that these memories are extremely empowering because once you overcome something for the first time, you know you can do it again. In fact, these experiences have equipped us to face extremely challenging unknowns with the placid understanding of how it feels to persist and succeed. It is this understanding of ourselves, of each other, and of the world around us that give me all the confidence that whatever we do next, it will be great. Even sometimes now, I find myself pinching my own arm, uncertain if this dream of mine has really come true. And there will be moments when you will too. But right now is not that moment. Right now, we radically celebrate this accomplishment with every single cell in our bodies, all 40 trillion of them. Thank you for your curiosity. Thank you for your perseverance. And thank you for all you will accomplish.
Isla, that was so beautiful and so moving and resonated with, I think, all of us. So thank you for those wonderful words. I'm very pleased now to introduce Dr. Wendy Garrett, who has the difficult task of reading out the names and dissertation titles of each graduate as they are hooded. Dr. Garrett is the Irene Hines Given Professor of Immunology and Infectious Diseases in the Department of Immunology and Infectious Disease and of Molecular Metabolism at the Harvard Chan School of Public Health, a professor of medicine at Harvard Medical School, and most importantly for me, the Associate Director of the PhD program in Immunology. Wendy?
Are they going to line up now? Do you want me to do the first one? Very exciting. Bioinformatics and Integrative Genomics. Joyce Blossom Kang, advisor Sumya Roy Chattery, Integrative Analysis of Large-Scale Single-Cell Genomics Data in Immune-Mediated Diseases. Benjamin Kompa, advisor Andrew Beam, New Methods for Representation Learning, Uncertainty Quantification and Causal Inference in Biomedical Machine Learning. Uparna Nathan, advisor Sumya Roy Chattery, integration of multimodal single-cell data to characterize T-cell phenotypes, gene regulation, and disease associations, and a certificate in leader human biology and translational medicine. Vinayak Venkatasesha Viswanandam, advisor Peter Park, origins, patterns, and consequences of somatic mutations in engineered normal and diseased cells. And a certificate in leader human biology and translational medicine. Of course. Maximilian Boss Thomas, advisor George Church, synthetic genome construction techniques to edit genetic code of Escherichia coli. Biological and Biomedical Sciences, Samuel Barrett, advisor Christian Dibble and Alex Toker, regulatory mechanisms linking coenzyme A synthesis and PI3 kinase signaling, and a certificate in leader human biology and translational medicine. Jonathan Bushman, advisor Eric Fisher, mass spectrometry-based proteomics approaches to define enzyme-substrate relationships in the ubiquitin proteasome system. Anita Chundrahus, advisor Joseph Arboleda, Reelin structure and interactions relevance to Alzheimer's disease. Cindy Xinyu Chang, advisor Scott Kennedy, regulation of transposon mobilization and C. elegans. Leon Chon, advisor Bill Sellers, identification and characterization of novel cancer genetic dependencies. Jihoon Choi, advisor Michael Greenberg, the activity-dependent phosphoregulon in stem cell-derived human neurons. Vicky Chow, advisor Tracy Young Pierce. Defining the relationship INPPD SHIP1 regulates inflammasome activation in human microglia, and a certificate in Leader Human Biology and Translational Medicine. Nicoletta Commons, advisor Maha Farhat, population genomics of the emerging pathogen Mycobacterium abscessus. Juliana Carrar-Fried, advisor Anna Greca, Falcon, systematically integrates free fatty acid biology and identifies new mediators of lipotoxicity. Anastasia Urshova, advisor William Scheer, engineering cooperativity for the control of biomolecular self-assembly. Michael Florea, advisor Amy Wagers and Luke Vanderberg, engineering whole body gene therapies for aging. You're going to do great. Olivia Foster Rhodes, advisor Angela DePace, yes, and looking beyond patterning genes and minimal enhancers in Drosophila segmentation. Patrick Griffin, advisor David Sinclair, improved methods for age, frailty, and mortality prediction in large-scale longevity studies. Dennis Grishin, advisor Alexander Gusav, allelic imbalance of chromatone accessibility in cancer identifies candidate causal risk variants and their mechanisms. Jeremy Gongabison, advisors Michael Pate and Ralph Weisleiter, neutrophil heterogeneity in cancer. Jimmy Guo, advisor, Andrew Aguirre, cell states and therapeutic targets in pancreatic cancer. Rahul Gupta, advisors Vamsi Mutha and Ben Neal, nuclear genetic control of mitochondrial function and its contribution to human disease, insights at biobank scale. Sung-Hoon Han, advisor, Eliezer Van Allen, investigating the landscape of rare germline variants in cancer from cancer risk to treatment response. Talia Hart, advisor, Alexander Sukas, Metformin action on nuclear transport. Alessandra L. Hillis, advisor, Alex Toker, exploiting signaling signatures in triple-negative breast cancer to identify targetable vulnerabilities, and a certificate in leader human biology and translational medicine. Sungwon Kim, advisors Jonathan Seidman and Christine Seidman, functional assessment of human variants and non-coding regulatory sequences. Shoa Kim, advisor, Anna Greca, dissecting the molecular mechanisms of cellular injury in the kidney. Karalampia Kutsiumpa, advisor, David Ginty, skin type dependent development of mechanosensory neurons. Mark Lipstein, advisor, Wesley Wong, high throughput functional screening and DNA nanoswitches. Patrick Loy, advisor, Karen Chakowski, epigenetic-based therapeutic strategies to drive differentiation and death in KRAS mutant colorectal cancers. New Win, advisor, David Fisher. The DDI2 inhibition promotes MHC class 1 antigen presentation and immune checkpoint blockade efficacy in cancers. Doigu Pezan Dogru, advisor, Jessica Whitted. Insights into systemic responses to amputation injury in axolotl salamanders. Alex Plesa, advisor, George Church, age reversal by transcriptomic reprogramming. Kristen Chan, advisor, Segal Kadosh, Dynamics and Spatiotemporal Organization of Mammalian SWE/SNF chromatin remodeling complexes and transcriptional regulation. Laura Christine Roche, advisor, Raoul Mostaslavsky, characterizing the transcriptional and chromatin landscapes of cancer metastasis. Ian James Roney, advisor, David Rudner, transport and coordination of membrane lipids during bacterial cell envelope biogenesis. Christopher Rota, advisor, Rosalind Siegel, astrocytes promote pediatric glioma progression and enhance ex vivo tumor modeling. Bing Shui, advisor, Kevin Haggis, marker RNA-mediated response to pathophysiological stress in the intestine. Chad Brandon Stein, advisor, Karen Edelman, promoter proximal transcription termination by the integrator and resoxysome complexes. Georgia Sturtz, advisor, Leonard Zahn, cellular and genetic dynamics of the melanoma microenvironment, and a certificate in leader human biology and translational medicine. Kartong Tan, advisors, Matthew Meyerson and Hang Lee, delineating genome alterations in cancer with long-read and linked-read sequencing. Samuel James Watrous, advisor, Leonard Zahn, on the role of the stem cell niche in hematopoietic development. Brandon Joseph Woods, advisor, David Van Vactor, the role of microRNA in activity-dependent synaptic plasticity. Hanxiang Zhang, advisor, Bruce Bean, the cannabidiol modulation of ion channels and neuronal excitability. Hanchen Zhao, Advisor Michael Eck. Biochemical characterization of the EGFR exon 20 insertion variants and their inhibitor sensitivities. Biophysics. Hans Bergel, Advisor Wesley Wong, using mechanical force to study biological processes from blood clotting to immune cell adhesion. Daniel Greenfeld, Advisor Connor L. Evans, Quantitative and Computational Approaches in Translational Biophysics Applications. Sophia Liu, Advisor Fei Chen, Spatial and Temporal Dynamics of Immune Cell Interactions. Sadie Payette, Advisors Joseph Laparo and Hari Arthonari, Disordered Proteins Regulate Stress and Damage-Dependent DNA Binding. Joseph Sedlak, Advisor Omer Yilmaz, The Role of Prostaglandins in a High-Fat Diet in Cancer Progression and Metastasis. Leila Siraj, Advisors Eric Lander and Hilary Finucane, Characterizing Regulatory Elements and Non-Coding Variants in the Human Genome. Chemical Biology, Megan McCurry, Advisor Sloan Devlin, Gut Bacterial Metabolism of Corticoids and Other Host-Produced Steroids. Sarah Cheyenne Potter, Advisors Stephen Haggerty and Suzanne Walker, Impact of Structural Alterations on the Cellular Functions and Interactions of Oglicnac Transferase. Immunology. Jenna Lynn Collier, Advisor Arlene Sharp, PD-1 function on T cells and spontaneous and anti-PD-1 mediated autoimmunity. Sophie Stephanie Berube-Jaguer, Advisor Facunda Batista, Codon Bias in tRNA Adaptation and Antibody Production. Catherine Jacob Dolan, Advisors Dan Baruch and Aaron Schmidt, Humoral Immune Response to COVID-19 Vaccines in Non-Human Primates and Humans. Kenta Musalenejaj, Advisor Jonathan Kagan, Organelle-Associated Innate Immune Responses to Self-DNA in Mammals and a certificate in Leader Human Biology and Translational Medicine. You got it. Alexander Schnell, Advisor Vijay Kutru, Th17 cell heterogeneity and tissue inflammation. Jeroen Martin Johannes Taas, Advisor Facundo Batista, modulation of B-cell responses by previously elicited antibodies. Catherine Ann Tooley, Advisor Anna Anderson, investigation of novel regulators of CD8 positive T-cell function in cancer. Sarah Weiss, Advisor Arlene Sharp, deletion of an exhaustion-specific PD-1 enhancer modulates CD8 positive T-cell fate and function. Omer Khalid Yagi, advisors Diane Mathis and Christophe Benoit, uncovering stromal cell functions in acute and chronic muscle injuries. Neuroscience. Melanie Bosnock, advisor Rachel Wilson, uncertainty and learning in biological ring attractor. Isla Bastille, advisor, Lisa Goodridge. A combinatorial MAF transcription factor code establishes synaptic heterogeneity critical for normal hearing function. Michaela Ennis, advisor Justin Baker. Behavioral quantification is the missing link between fields. Tools for digital psychiatry and their role in the future of neurobiology. Daniel Gillum, advisor Michael Greenberg. Synaptic activity couples inducible transcription to genome preservation in the mammalian brain. Xiao Meng Han, advisor Jeff Lickman. New methods of correlated light and electron microscopy provide a multimolecular overlay for large volume connectomic data in the brain. Lingfeng Ho, advisor John Assad. Substantia nigral activity and self-timed movements. Matthew McGill, advisor Daniel Poley. Cortical plasticity mediates behavioral hypersensitivity following noise-induced sensorineural hearing loss. Si Zhang, Advisor Christopher Harvey, Organization of Neuronal Representations in Mouse Posterior Cortex for Dynamic Navigation Decisions. Tsihi Alex Chang, Advisors Clifford Wolf and Jan Drogoswich, The Rodent Body Language of Ongoing Pain. Adrian Cho, Advisor Gerald Kidd, Speech and Hearing Bioscience and Technology. Title, Auditory Motion Perception in Multiple Sound Source Environments. Wendy Chen, Georgian, Advisor Tiffany Hogan. What's in a Name? Investigating Labels and Legislation in Education for Children with Developmental Language Disorder. Sarah Goots, Advisor Jordan Green, Automatic Speech Recognition as a Clinical Tool, Implications for Speech Assessment and Intervention. Tanya Talker, Advisor Thomas Quadieri, Detection and Characterization of Autism Spectrum Disorder and Parkinson's Disease using measures of speech and fine motor coordination. System, Synthetic and Quantitative Biology. Haley Cambra, Advisor Alon Klein, Tissue Intrinsic Properties of Regeneration and Homeostasis in Injury in the Mammalian Small Intestine. Tanish Jagdish, Advisor Andrew Murray and Michael Desai, The Dynamics of Fitness and Pleiotropy in a Long-Term Evolution Experiment with Escherichia coli. Maximilian Gabriel Marin, Advisor Maha Farhat, peering into the dark matter of the M. tuberculosis genome using long-read sequencing. Jackson Naiman, Advisor Eliezer Van Allen, Spatially Aware Deep Learning for Clear Cell Renal Cell Carcinoma Characterization and Discovery. Suyang Wan, Advisor Johan Paulsen, Development of a High-Throughput Image-Based Single-Cell Genotype-Phenotype Mapping Platforms. Virology. Camille Hoyle Cushman, Advisor James DiCaprio, understanding the requirements of PRMT5 splicing regulation in Merkel cell carcinoma. Thomas Charles Frost, Advisor, James DiCaprio, Molecular Subtyping and Vulnerability Exploitation in Merkel Cell Carcinoma. Sumi Lee, Advisor, Frank John Slack, role of host microRNAs in Kaposi sarcoma, herpes virus, tumorigenesis, and lytic reactivation. Austin Hurst Mani, Advisor, Max Nybert, discovery and characterization of a novel double-stranded RNA virus infecting the human parasite Trichomonas vaginalis. Adam Nitido, Advisor, Alejandro Balas, the role of mutation rate in HIV diversification and adaptation in vivo. Meredith Phelps, Advisor, Alejandro Balas, broadly neutralizing antibody recognition of diverse HIV envelopes modulates effector function. Paul W. Rothloff, Advisor, Sean Whelan, viral and host determinants of cell entry of emerging and neglected enveloped viruses. Kaylee Madison Slavik, Advisor, Philip Kransich, see gas-like receptors in animal innate immunity. Catherine Noble-Sadrowski, Advisor David Knipe, host cell determinants of interferon control during herpes simplex virus infection. Last but not least, Stephanie Payton Yi Yu, Advisor Benjamin Gewurtz, Epstein-Barr virus, evasion of intrinsic and innate immune pathways during the viral lytic cycle.
Thank you, Wendy, and congratulations to all the graduates, and let's have a round of applause for them again. Okay, thanks, everybody. So it is now my great pleasure to introduce and turn this over to Emma Dench, the Dean of the Ken Griffin School, Graduate School of Arts and Sciences at Harvard University, and the McLean Professor of Ancient and Modern History. So, Emma.
That was incredible. Congratulations. Well, what an honor. It feels as if I spent the whole day with many of you because from the morning to Sanders Theatre. But Sanders Theatre, it was lovely, but so much more superficial than what we just witnessed. And I just can't tell you, I love this ceremony, the hooding ceremony at the Division of Medical Sciences. And I love two things about it very, very much. As what Ros was saying, I'm a humanist, right? So I'm the person from Mars. And the two things I really, really love about this ceremony are, first, I really, really like your long, long titles. I do have a bit of a complaint because I feel that there should be a little bit more Latin and Greek because that's what makes me feel smug because I can understand it. But I'm actually serious. The very, very long titles just give me so much insight into how you found your own completely unique path and how it relates to others. And I know that I can just detect that very superficially, but I know that every word is coded about what your individual contribution was and how it relates to other people's. Wow, and I just love it. It's like, it's music. And the other thing that I really, really love is the actual hooding. And I was going to use the very old-fashioned, slightly biblical terminology of laying on of hands. Is that all right? But just the actual, both the physical hooding and even actually much more than that. Many of you, it's a group activity. And again, I come from such a solitary business, but for me still, even coming from such a sort of solitary discipline when we're very, very lonely, advising, it's all about doing research, it's all about teamwork, it's about the village. And every group that I saw up there with the laying on of hands, again, if you don't mind me saying, just spoke volumes to me about these are people who've been with you. You've probably been with each other more than you think. It's probably been more reciprocal than particularly as students you even imagine. But you've been together thick and thin through the really, really dark times and the celebrations, and I find so much joy in that. So those are just my 2 cents, but warmest congratulations. What a privilege to spend much of the day with most of you, and please celebrate. And we're just so enormously proud of you. I think I can speak for everybody in the room. Congratulations, you're fantastic.
Thank you, Emma, for your really inspiring remarks. Like you, one of the things I really love about the hooding is that it's such a wonderful ceremony, and not only does it involve the village, but it's also connecting the past and the future with the advisor and the advisees. And so it's really been wonderful to spend this time with you. Finally, before we finish, I want to thank several people who are not on the stage and have been so instrumental to the education and support of our students. I want to thank Sam Reed, the Executive Director of the Hills Program, and the wonderful faculty leaders, program administrators of all the programs. I want to thank all the faculty mentors who have meant so much to you all and have really been involved in such a important relationship, both for the faculty and for you. Finally, I want to thank Nancy Gerard and the team from the Office of Graduate Education who organized today's events. The nice thing about this hooding is it's a little more casual, but they make it all work so beautifully. So thank you. Finally, I'd like to ask the audience to please allow the graduates to leave in a procession first as we all move to a reception to honor the new graduates. And thank you all for joining us today. It's really been a pleasure. So again, congratulations.